Hello everyone, this is Rohan Mishram and welcome back again on my lecture series on bioinformatics. So in our previous video, in our previous lecture, we have discussed some important terms and concepts in molecular phylogeny. We have discussed different terms like types of homologs, what are orthologs, what are paralogs, and how do they arise. Okay, and today we would discuss the uh, uh, today we would discuss what are the important tools and what are the important techniques using which you can reconstruct a tree or phylogenetic tree. Okay, so to begin with, uh, modern phylogenetic analysis, you can say that it has got two aspects. The first aspect would be generating an effective multiple sequence alignment of a given set of sequences, and these sequences can be protein sequences or these sequences can be amino acid sequences as or protein sequences or nucleotide sequences as such. Okay, and once you get hold of this multiple sequence alignment, the next thing that you are supposed to do is to use this multiple sequence alignment as an input for generating phylogenetic tree, and the process by which we generate such phylogenetic tree is called as tree reconstruction. So this is how usually the protocol is. First, we generate multiple sequence alignment of given sequences, and then we try to reconstruct a tree that would give us an idea or give us some insights on how these sequences might have evolved. Okay, so major focus of our discussion today would be tree reconstruction, as to how do we reconstruct the phylogenetic tree, what are different methods that are available, when to use what type of methods. This would be the major point of discussion of today's lecture. Okay, so now let us first try to summarize as to what are the different options that are available for us for tree reconstruction. Well, broadly, you can classify the tree construction methods into two categories. The first category, what we call it as the distance method, and another category that we have is called as character-based methods. So, character-based methods, again, there are two methods available which can be classified as character-based method. The first method is called as maximum parsimony and maximum likelihood. So, first, let us try to differentiate between what are these methods, right? And what are the features of these methods, and then we would see how, when, and how to use them, right? So, let us first try to distinguish between what are character-based methods and how do they differ from distance-based methods. Okay, so in this character-based methods, what we do is that we use the aligned characters such as your DNA or protein sequences directly during the tree inferences or tree reconstruction. The idea is that when we use character-based methods like parsimony or likelihood, okay, so when we use character based methods like parsimony and likelihood, we directly use data from the multiple sequence alignment or whatever character data that we have in our multiple sequence alignment. We use that and generate your phylogenetic tree as such. Okay, something like this. Using the characters information, we directly generate a tree using these character based methods like maximum parsimony and maximum likelihood. Okay, another type of method, what we call it as distance based method, in which we do not directly use the character data. What we do is that we convert this character data into pairwise distance, or what we call them as dissimilarities. So what we need to do is that using this multiple sequence alignment data, we generate pairwise distance matrix that accounts for the similarity or dissimilarity between the sequences that you are dealing with. And once we have this pairwise distance matrix that gives an account of how far or how similar or how dissimilar the two or three or whatever sequences are, then what we do is that we use clustering algorithms or clustering methods like neighbor joining and UPGME for tree reconstruction. So this is what it is done. You start out with sequences, multiple sequence alignment. You convert this multiple sequence alignment into, say, some matrix distance matrix or distance data. Okay, and this distance matrix would indicate the similarity or dissimilarity between the sequences, and using this data, then you use clustering algorithm like neighbor joining and UPGMA, and eventually you draw your phylogenetic tree. Okay, so this is the general procedure for using or for tree reconstruction using distance based method. And this is how you directly use the character information from your multiple sequence alignment and reconstruct the tree using character based methods like parsimony and maximum likelihood. So this is the basic difference between the character based methods and distance based methods. So now let us try to understand as to when do we use what type of method. Okay, so usually the very first step that you perform in phylogenetic analysis is to obtain the multiple sequence alignment. Well, this particular image, I have taken this image from the book Bioinformatics Sequence and Genome Analysis, written by David Mount. So it is one of the most standard book that is usually referred by bioinformatician for understanding concepts and for demonstration purposes as well. Okay, so as per this book, what we do is that we begin with multiple sequence alignment. Okay, and once you have your multiple sequence alignment, based on the data that is present in your multiple sequence alignment, you need to then make a decision as to what type of tree reconstruction method is needed to be implemented for drawing the tree or for the tree reconstruction as such. Okay, so what you do is that you ask yourself a question ki, if there is any strong sequence similarity or whatever sequences that you have aligned in multiple sequence alignment, do they have very strong sequence similarity? That means, can you find out most of the sequences, well, say for example above 90% quite very conserved in your multiple sequence alignment? If that is the case, then you are supposed to use maximum parsimony. Okay, so what do we say that what exactly do we mean when we say that there is strong sequence similarity? That means, well, if in your multiple sequence alignment there are say some 100 columns that are present in your multiple sequence alignment, of these 100 columns, if there is a high level of conservation in more than 95 columns, then we say that there is strong sequence similarity in the given multiple sequence alignment. Okay, so if that is the case, then you are advised to use this maximum parsimony method. Then if you get a negative answer to this question, okay, that there is no strong sequence similarity observed in your multiple sequence alignment, then you ask yourself another question ki, is there any clearly recognizable sequence similarity? Okay, well, are you in position to recognize that okay, of these 5 or 10 sequences, there are certain sequences where, so for example, there are 5 sequences in uh, my multiple sequence alignment out of 7 that are highly similar, while these 2 sequences are dissimilar. Okay, if that is the case, then you are advised to use this distance based method. Okay, so if this is the case that some of the sequences are similar while some of the sequences are dissimilar in your multiple sequence alignment, then you are supposed to use this distance method. And still if you get the negative answer ki okay, in my multiple sequence alignment, leave aside strong sequence similarity, none of the sequences are similar, in that case, and but you are sure that these sequences are somehow related, then you are supposed to use this maximum likelihood method as the final resort. Okay, so whatever method you use for tree reconstruction, either you can use maximum parsimony, distance based methods, or maximum likelihood method based on how is the sequence conservation that is observed in your multiple sequence alignment. You at the end of these methods, you would be able to generate a tree, okay, by using any of the method, distance based method or character based method. Okay, and finally what you are supposed to do is that you are supposed to, after tree reconstruction, you are supposed to analyze how well your data support your prediction. Okay, so this is, you know, a typical protocol that is followed when you perform phylogenetic analysis. Okay, based on the sequence conservation and the number of sequences that are present in your multiple sequence alignment, you would select an appropriate tree reconstruction method. That can be either character based method or that can be either distance based method. Okay, so well, now let us try to uh, discuss in very short brief as to what are different features of these tree reconstruction methods. Okay, so when do we use parsimony now? As I have already mentioned, you can use parsimony when you figure out in your multiple sequence alignment that not very different sequences are present and the variation is very small in your sequences. Then you are supposed to use this maximum parsimony. Okay, so there is also a limitation of how many number of sequences should be there if you are using maximum parsimony, right? So we'll say, for example, this method is quite suitable if you have less than a dozen of sequences for tree reconstruction. Okay, now if the number of sequence exceeds 11 or 12, this parsimony method may not be very suitable. Okay, why? Because if there are more number of sequence, that would mean that it is possible that this parsimony method would generate equally parsimonious trees, multiple equally parsimonious trees. Okay, and it would then become difficult for you to recognize that out of these 3 or 5 equally parsimonious tree, which one is the better one. So if that is the case, in such case what you do is that, well, say for example, you start out with multiple sequence alignment, right? And using character based, and in your multiple sequence alignment, now you know that there is the sequences are quite well conserved. Okay, and very small variation is present in your sequences. Then you use maximum parsimony, and what you do is that you use this maximum parsimony method and generate a number of trees. This is tree number 1, we'll say, for example, and you have generated tree number 2, right? And this is tree number 3 that you have generated, right? So now it is entirely possible, as the number of number of sequences increases, if however if they are very sequentially conserved, at the end of this parsimony method, what you would do is that you might end up with tree number 1, tree number 2, tree number 3, which is equally parsimonious. And in such a case, what you do is that you try to then generate a consensus tree out of these three that would really correspond a consensus of all these three, and this would be your final final parsimony or parsimony based tree. Okay, so this maximum parsimony is based on Occam's razor principle. Okay, and as per this Occam's razor principle, well, it is assumed that whatever multiple sequence alignment or tree reconstruction that you would generate, it is produced, or it is a product of minimal amount of mutations or minimal amount of variations that are needed to be considered for tree reconstruction. Okay, so that is the major principle behind using this maximum parsimony. Okay, so now if you tend to use the sequences, or you can say the multiple sequence alignment, where variation is quite very high. Okay, so that variation in this way might be that variation might result into many possible ancestral relationships, and thus you might end up with say some very erroneous number of trees. That would be very difficult for you to recognize which one is the most maximally parsimonious tree. Okay, so these are the features of the character-based method called as maximum parsimony. So don't worry if you don't understand much of the principle behind this. So in practical point of view, when are you supposed to use maximum parsimony? When the variation is very small, that means the sequences are quite very conserved in your multiple sequence alignment. Okay, and the number of sequence is less than 12 or around 12 or 13. Okay, so if that is the case, then you are very free to use maximum parsimony, right? Then another method that we use is something called as the distance method, and these distance methods are able to predict evolutionary tree when variation among the sequence is present. That is, some sequences in your multiple sequence alignment, we'll say for example we have this multiple sequence alignment, some of these sequences are similar while some of these sequences are dissimilar. That is, some sequences are more alike than others. If that is the case, then it is advised that you use this distance based method, right? So when the amount of variation is kind of intermediate, okay, not all the sequences are quite highly conserved, while some of the sequence can be variant or divergent, right? If that is the case and you have big number of sequences, or what we can say large number of sequences are there in your data set, then you usually use this distance based method. And many of the time this would be an ideal situation, that you might end up with a large number of data set, huge number of sequence might be there in your data set, and if there are huge number of sequence, then there is possibility that few of the sequences can be very similar to each other, while some of the other sequences cannot might not be very similar. Okay, and in such a case people usually end up using distance based method. Okay, so distance based methods are very popular in tree reconstructions. in most of the papers, or in most of research papers, or most of the books, or most of the seminar, you might have heard that people normally use this distance based method, right? This is the basic reason behind it. Okay, most of the time it happens that you end up with, say, some 50 or 60 or 100 sequences, and if that is the case, then distance using distance based method becomes handy, and it becomes quite optimal to be used, right? So this is how you are supposed to use these distance based methods. When the variation among the sequence is present, that is intermediate conservation is there, right? And you have a large number of sequences to handle, right? If these two conditions are met, then you are supposed to use this distance based method. Okay, okay, now the last one that is remaining, okay, what we call it as maximum likelihood method. So this maximum likelihood method may be used for any set of related sequences. Okay, well, say for example, you end up with multiple sequence alignment in which now you are in a situation that all of these sequences are not quite very conserved, or you can say the variation is very high. Okay, so if the sequences are quite variable, okay, that is they don't share much of the conservation in their sequence. Okay, so this is the particular case when you would go and use maximum likelihood method. Okay, so it is particularly useful when the sequences are more variable. Okay, these methods, what we are talking about is maximum likelihood methods, are computationally quite very intensive, and the complexity of the computation increases when the number of sequences increases. Okay, you can get the results quite very easily when some small amount of sequences that are variable are provided as an input to maximum likelihood method. Okay, but the biggest advantage of this method is that they would provide some evolutionary model to account for why there is variation among the sequences that you have given. Okay, it would account for the variation that is present in your sequences. Okay, it might provide some evolutionary models to do that. So this is how uh, you select an appropriate method for for proceeding for reconstruction or tree reconstruction as such. Okay, now let us start discussing on the fact that, well, say you have kind of created a tree from your multiple sequence alignment by whatever method, by maximum parsimony method or maximum likelihood method, or you might have used distance based method. Then you might ask yourself questions like, is my tree really very reliable, or how can I decide if the tree that I have generated is reliable or not? Okay, in order to answer this question, people usually implement this bootstrap analysis. Okay, the main principle or the main procedure in bootstrap analysis is that you first resample your multiple sequence alignment, and for each resample data set you would construct a separate tree, and after, in this way, for each separate data set you would end up with multiple trees. Okay, and at the end of this step you would assign some reliability weights on each of the internal branch of the inferred tree. Okay, so and based on these reliability weights, then you can decide ki which part of my tree is more reliable as compared to other. Okay, well, say for example, at the end of this bootstrap analysis, you might end up generating a phylogenetic tree okay, like this. And at each of the internal node, you might end up assigning some reliability weights. Well, say for example, if you have generated say some 100 trees from 100 resampled multiple sequence alignment, and out of these 100 times it was observed that 95 times the sequence A and B were found to be present together. So in this way, you can now say that this much portion of my phylogenetic tree is more reliable. Okay, the reliability of A and B being together is very quite high. So this is how you perform this bootstrap analysis and determine the reliability of your tree. So things would be more clear in this slide. So when you begin with your multiple sequence alignment, okay, this is your original data set. Okay, and we'll say we would now call each and every column with say some number. This is 0th column, first column, third, second column, third column, and so forth. Right. So this is your original data set. In bootstrap analysis, what we do is that we reshuffle the columns in your original data set. Okay, by reshuffling, what I mean is that you would transfer the first column to fifth, and seventh column to second, and third column to ninth, and ninth column to sixth, so on and so forth. Okay, so by reshuffling, what you would of course end up the sequence of your multiple sequence alignment would be different, but eventually the variation that is present in your multiple sequence alignment would remain the same. Okay, so in this way, what you do is that you reshuffle the columns in your multiple sequence alignment, and you would generate different replicates of your multiple sequence alignment. Well, say for example, over here, you can see that after reshuffling the columns, what you have is first, fifth, sixth, and second and third column in this way. We would call it as replicate 1. Similarly, after reshuffling, we would generate replicate 2, replicate 3, and so on and so forth. So in this way, what we say that we have assembled some pseudo data sets. Okay, and we would repeat this reorientation or rearrangement of your multiple sequence alignment data set, will say for example 1,000 times. So in this way, we would generate say some 1,000 pseudo replicates or 1,000 pseudo data set that would correspond to your multiple sequence alignment in 1,000 ways. Okay, so from each replicate we would generate a tree. From replicate 1 we would generate tree 1, from replicate 2 we would generate tree 2, from replicate 3 we would generate tree 3, and we would thus generate 1,000 trees from these 1,000 replicates. Okay, so the idea is to build tree for each pseudo data set that you have generated, and in this way you would end up generating say some 1,000 trees. Okay, so next step would be to find out a consensus tree of these 1,000 trees that you have generated from this 1,000 pseudo data set that you have created. Okay, and after generating a consensus tree, you would assign some reliability weights at the internal nodes, right? So this would indicate that out of these 1,000 tree, 67% of time the sequence A and B were found to be together. Okay, or this might happen at this particular node 98% times the sequence A, B and C were found to be present in a single clade. So now you can say that the reliability of this sequence A, B and C to be present in this particular arrangement is very high, right? So in this way, after constructing a very strict consensus tree and based on the reliability weights that you have obtained at the internal nodes, you can define the reliability of your tree. Like what part of my phylogenetic tree is more reliable than some different part of your phylogenetic tree. Okay, so this is how you perform the bootstrap analysis. Okay, so now how would these things practically work, right? Well, say for example, you might generate multiple sequence alignment using software like Clustal. Okay, and if you are using a computer program or a set of computer program called as Phylip package, okay, then this is how you might work. You begin with multiple sequence alignment that you might obtain using softwares like Clustal. Then in this Phylip package, you have a computer program called as sequence boot that would generate such 1,000 different pseudo data sets. Okay, so you might end up with say some 1,000 multiple sequence alignment. And now based on if your multiple sequence alignment is from protein, and you end up identifying that I would be using this distance based method. Okay, so first you would use computer programs like protdist if your sequences are protein sequences, and you would then generate 1,000 distance matrices. Now if your multiple sequence alignment is made up of nucleotide sequences like DNA or RNA, then if we'll say for example if it is made up of DNA sequences, then you would use DNA dist as a computer program that would generate 1,000 distance matrices. Okay, so once you generate those distance matrices, you would use clustering algorithms that are implemented in programs like neighbor, fish, and cache. Okay, so using these these clustering algorithms that implement either neighbor joining algorithm or UPGMA. So based on these softwares, you might end up creating 1,000 phylogenies from 1,000 distance matrices. So once you end up with 1,000 phylogenies, you can use another computer program called as Consens, which is part of this Phylip package, and generate a single phylogeny. Okay, and this single phylogeny would be in your Newick tree format. You can use this Newick tree format as an input to this drawgram program, which is also a part of this Phylip package, and create final rooted tree. So this is how you perform distance based analysis using your bootstrap analysis for defining the reliability of your tree. Okay, then if you want to use something, if you don't want to use distance based method and you want to use character based method, so what you would do is that you would begin with multiple sequence alignment, then use the sequence boot, create these 1,000 multiple sequence alignment, 1,000 data set, and based on what type of character based method you are using, you have appropriate programs in your Phylip package, right? So if you are trying to use character based method like maximum parsimony and you have protein sequences in your multiple sequence alignment, then you would be using protparse as a computer program. If your multiple sequence alignment contains DNA sequence and if you want to use character based method like maximum parsimony, then DNA parse would be the computer program that you would be using from the Phylip package. And if you are trying to generate uh, a tree using character based program like maximum likelihood, and you end up with working with protein sequences, then you can use these two computer program, ProMLK and ProML, for generating phylogeny. If you are using DNA as your DNA in your character data set, then you can use DNA MLK and DNA ML for generating the phylogenetic tree using maximum likelihood method. Okay, so whatever computer program you use, you might again end up with 1,000 phylogenies, creating 1,000 trees, right? And from again these 1,000 phylogenies, you can use this consensus program to create a single phylogeny, which would be in Newick format. Again, you can use programs like drawgram, or there is another program called as draw tree that can be implemented to generate unrooted tree. This drawgram is used to generate rooted tree. Okay, so this is how it practically works when you use programs like Phylip. So it is not like Phylip is the only package for generating multiple sequence alignment. There are other tree reconstruction packages that are available. Mega is another software that is popularly used nowadays, right? But in our demonstration that is going to happen after this lecture, we are going to use Phylip as a package for tree reconstruction. Okay, so we would be using these different programs, right? We would be using sequence boot computer program to generate 1,000 multiple sequence alignment pseudo data sets. Then we would be using prot dist to generate 1,000 distance matrices. We would be using neighbor program to generate 1,000 phylogeny. Then we would be using consensus program to create a consensus tree from these 1,000 trees that we have generated. And after analysis of this consensus tree, we would be using drawgram to generate a final rooted tree. So this is what is going to happen in your demonstration section. Okay, now let us move on to discuss one of the method that is used to generate a tree from the distance matrices as such. Okay, or we would see how this neighbor joining clustering algorithm works. Okay, so now to begin tree reconstruction using this neighbor joining method, well, it is assumed uh, that the tree it is assumed that there is just one internal node from which uh, all the branches are radiating like star like pattern. Okay, so this is the starting point of your neighbor joining algorithm. We would assume that all of the sequences are originated from a single ancestor. Well, this is a wrong assumption, but we have to start from some place. Okay, so this would be the starting point. The starting point, we would assume that all of the sequences, whatever we are dealing with, they have originated from a common ancestor. Okay, and this common ancestor is P, and they radiate from a star like pattern. Okay, so now next thing what you do is that in the next step you choose any two random sequences. Okay, so in this case we have selected randomly sequence C and sequence D. Okay, so what you do next is that after randomly selecting any two sequences, what you do is that you would remove these two sequences C and D from the original cluster, and you would attach them to an internal node, and we would call that internal node or ancestor as Q. Okay, what we do is that we remove any two randomly selected sequences from the star, okay, which have this center P, and after removing we would attach these two sequences to a second internal node called as Q. Okay, now this second internal node is again connected to the branch, or you can say the branch to the center of your star. Okay, so next what we do is that we calculate what is the branch length of this new tree. Okay, we would calculate the branch length of this new tree, which is composed of C, Q and D, right? So this is what we do. So randomly we select C and D, and calculate the branch length. So this is what we are doing. We are having this queue, okay, and we have now selected randomly C and D, okay. We have added it to the queue, and this queue is again connected to the center of your star where other sequences are there, right? So after doing this, what we do is that we calculate the branch length of this C, Q and D. We'll say, for example, we ended up with calculating the branch length as B1. So again what we would do is that we would again place this C and D back to your star, okay, and then again randomly select some another two sequences. Well, let's say for example, now we have ended up selecting A and B, okay. So this would be the case. We would take this B, A and B out of your star, attach it to Q, right, attach it to Q, okay. Over here what we have, A and B, and again calculate the branch length. We'll say for example, now the branch length is B2, okay. We would again replace this A and B back to the star, again select any two random sequences H and G, take them out, okay, and connect to this Q. Over here we have got now H and F. Again we would calculate the branch length. We'll say for example, the branch length that we have calculated now is B3. So in this way what you do is that you randomly select any two sequence and take them out and attach to this second internal node Q and calculate the branch length. In this way you would do the same unless and until all of the sequences have been considered and all of the branch length have been calculated, okay. So at the end of this step you would have different branch length 1, B1, B2, B3, B4, B5, so on and so forth till Bn, right? So these branch length indicate the uh, uh, these branch length indicate the different combination of the sequences that you have been trying out, okay. So of these branch length that you have, you would select or you would try to figure out which one is the lowest, okay. So we'll say for example, if you end up identifying this B4 as the lowest number in all the calculated branch length, what you would do, and we'll say for example this B4 correspond to the branch length of A and B, right? So this is the shortest branch length, okay. So what you would do is that you would end up identifying this A and B as the final neighbors in your phylogenetic tree, and you would permanently attach it to the second internal node, okay. So this would be the final neighbors in your phylogenetic tree, right? So these two sequences are combined into a single unit, creating a new star, and you would repeat the process of pair selection and branch length calculation and locate the second pair. The second pair would be again added to the first internal node as R, and you would again go on repeating the same procedure. Whatever sequences that are remaining in your star now, you would again randomly select two and calculate the branch length, then again select these two and calculate the branch length B2, then select third one, calculate the branch length B3. You do at this location now pair selection and branch length calculation for the remaining sequences in your star, okay. And whatever branch length now at this first iteration, you would find out whichever is the lowest branch length, you would use that branch length and the corresponding sequences as the neighbor or the next neighbor. So in this way you would go on iterating this step of pair selection and branch length calculation and select which pair is having the lowest branch length, okay. So in this way At first iteration you have separated or you have identified one of the pair of sequences that would remain final, or you can say the final neighbors in your phylogenetic tree. In second iteration you would identify another pair. In third iteration you would identify another pair. In this way you would go on. At the end of third iteration you would end up generating a phylogenetic tree as this, as shown over here. Okay, so this is how your clustering algorithm works. The clustering algorithm that would use data from your multiple sequence alignment and eventually the data from distance matrix. Okay, so in this way you generate complete reconstructed phylogenetic tree. Okay, so I hope you understand what how this neighbor joining algorithm works, right? So now let us talk about this simple package called as phylip. Okay, and this phylip is a very popular software that is used for generating this phylogenetic tree. Phylip is a package, or you can say a software for phylogenetic analysis, and since it is a package, it is composed of multiple programs. Okay, so this is the most widely distributed phylogenetic analysis package. Okay, so since it is a package, it contains a set of 30 programs that can be implemented in different phylogenetic analysis algorithm. Okay, so for performing character based method, and if we'll say for example in this package what you have is that if you want to perform character based phylogenetic analysis and you have DNA as a multiple sequence alignment data set, then you can use DNA parse for performing maximum parsimony analysis. If you have got protein as your data set, then you can use prot parse. Okay, this way there are how many 30 programs that are present in your phylip package. Okay, and this 30 program implements different phylogenetic analysis algorithm like maximum likelihood, like distance based method or parsimony based methods. Okay, now each of the program can be run separately in command line environment. Okay, and it is very simple to run this. All you need to do is to double click and the command line version of the program would appear. Okay, so by default most of the program would look for an input file called as in file, and they would write and output file called as out file. So whatever programs that are present in your phylip package, they would look for an input file called as in file, and after performing their function or after performing or after calculating a tree, they would generate out files as such, and these out files can be, you know, visualized by the user for phylogenetic analysis results. Okay, so now phylip is most flexible package and the programs can be used together in many ways. Okay, for example, to analyze a set of protein sequences with phylip, one may follow one of the multiple path. One of the path can be is that if you begin with protein sequence alignment using you can use, if it is a protein you can use prot dist and create a distance matrix. Once you have this distance matrix, you can input this distance matrix into neighbor and generate a phylogeny based on neighbor joining, right? Then you can use the output of this neighbor program and read the phylogeny into draw tree and produce a unrooted tree, right? Or the other way around, you can use it use like you can read the multiple sequence alignment using prot parse, produce a phylogeny based on parsimony. Okay, once you have a parsimony based tree, you can use this drawgram and generate a rooted tree. So there is flexibility of using these computer programs. It is not like it is to be used in a specific way. You can mix and match according to your need, right? So the thing is that each of the phylip program is very exhaustively documented in the doc files that are available with phylip distribution. Okay, so when you download the program, you also download their documentation. So this documentation has been converted into HTML by several groups so that you can visualize, or you can refer it online when you are working. Okay, so what you are supposed to do is that you are supposed to visit the phylip documentation page. You can download it. There are several organizations that have made phylip servers available. You can do the same stuff online as well. But in our demonstration, we would be doing it offline. Okay, so this is what I would like to conclude. We have learned what are the different methods that are available for tree reconstruction. Okay, we have learned that trees can be reconstructed using character based method or distance based method, right? So we have also discussed how and what might be the logic behind selecting a suitable method, when to use parsimony based method, or when to use distance based method, or when to use your maximum likelihood method. Okay, we have also discussed on as to how do we evaluate the reliability of tree. Okay, the tree reliability can be inferred using the bootstrap analysis. Okay, we have discussed one of the clustering algorithm that is used for tree reconstruction. We have discussed what is neighbor joining algorithm for tree reconstruction. Okay, and I have I have kind of introduced you uh, on how do we use popular software package for conducting phylogenetic analysis. So what would come next now? In our next lecture, we would begin demonstration of how do we perform multiple sequence alignment and tree reconstruction. Okay, we would be performing this multiple sequence alignment using cluster. We would visualize that multiple sequence alignment using GeneDoc, and finally we would try to construct a phylogenetic tree using different programs from the phylip package. Okay, so this is what we have done so far, and in our next lecture we would see how do we generate the phylogenetic tree practically, right? So see you guys in our next lecture.